Four, three, two, one. Good afternoon. This is Dr. John Bennett broadcasting from Miami. Today on Oncopedia TV, we're pleasure to have John Giannios, MD, a doctor from Greece, who is very prominent in the precision medicine field. Today, he's going to talk about precision cancer medicine in the postgenomics era. And we're also graced with the presence of two distinguished student panelists. So we'll start introduce them before we turn it over to John. Hello, Abdallah.
Hello, I'm an Egyptian medical student in Romania, third year medical student in Carol Davila University in Bucharest.
Very good. He's quite a video maker too. Okay, Slaven, please introduce yourself.
Hello everyone, my name is Slaven Gojkovic. I'm a medical student at the University of Zagreb and I'm very interested in neurosurgery, oncology. So it is a pleasure to be able to view this lecture today. Thank you.
Very good. And thank you for coming, John. It's all yours.
Thank you so much. Thank you. Okay, I'm going to talk today about personalized cancer medicine with molecular targeting and nanomedicine in the post-genomic era. I have done lots of cancer research concerning the establishment of precision cancer medicine based on omics, transcriptomics, nutrigenomics, epigenomics, pharmacogenomics, lipidomics, metabolomics, all kind of omics. So what I do is, after the genomic profile with RNA sequencing for coding RNA of each patient, you can do molecular targeting and facilitated by nanomedicine, you can tailor for even metastatic tumors. This is one of my works. It was chemoradioimmunotherapy for advanced breast cancer, and that was it was a novel technique to kill metastatic breast cancer cells by circumventing their chemo and radioresistant mechanisms. And I presented this in the European Association of Cancer Research, and went to the medical news today. That was in 2004. Many things have, taken place afterwards. There are some of my new therapies presented in NASCO. Letters are very small here, but there are quite many novel therapeutic approaches. And what they do, these approaches, I have used nanosomes to deliver adescent oligonucleotides. I have intervened in the epigenome by demethylating CpG islands and activating tumor suppressor genes, which used to be silenced, and they used to inhibit apoptosis, which is a very bad chemo-resistant mechanism. There are quite many novel therapeutic approaches. You can see them further down. I just put some of them because there are many. And some other ones in the American Association of Cancer Research. And all of these novel approaches, they aim to find on a personalized basis the unique genomic alterations which they cause chemoresistant and radioresistant and immunoresistant. And I try to circumvent this, and then with conventional therapy, again on a precision basis, I try to induce apoptosis to these chemoresistant tumor cells and radioresistant. Yesterday, I gave another approach, which was to eradicate cancer stem cells with nanosurgery. Today, I'm going to speak in general how we can eradicate tumor cells and how we can make them more susceptible to treatment. So there are some of these novel approaches which I have presented, and some other congresses too, in many other congresses too. And all of them, they are, as I said, tailored to the individual genomic profile of each cancer patient. That's the only way you can have some success. Okay, so that means we cannot have the blockbuster policy like the one size fits all. It does not apply to cancer. Cancer is a very heterogeneous disease, and especially metastasis, we have more than 100 different tumor types. So during my career, I have taken some awards. One of them was in San Francisco, and was a panel of Nobelists which they awarded this award concerning novel approaches against breast cancer. The specific one also has my daughter's name again. Inhibited oncogenic protein. Some proteins are quite chemoresistant. They cause great chemoresistance because we have coding RNA which is translated into these very chemoresistant proteins, like MDR1, for instance, which is a drug efflux pump. In this case, I have worked epigenomically, which means I have been in the non-coding RNA, which does not translate into protein, but what it does, one of these, for instance, microRNAs, one of these genes can control more than 1,000 of the coding RNA genes, which means that if we don't have an oncogene-dominant effect, something that can dominate all the other downstream signaling pathways, we cannot, I repeat, we cannot induce apoptosis. And as I said, apoptosis is what we need here. And as I explained yesterday, is the D2 stage, the reversible stage of apoptosis. I have written a few books concerning epigenomic medicine and genomic medicine and personalized medicine, and also I have studied many very bad tumors like glioblastoma, for instance, which is called the Terminator. So let me just make a review concerning this approach, the precision cancer medicine approach, based on omics and based on nanomedicine. Personalized cancer medicine versus blockbuster drug policy. Hippocrates, which he was our greatest doctor, and I think he's the greatest doctor ever, because his principles, they still exist, and he proves himself right all the time. He said that cancer, he had many cases of cancer, and he has been a basic proponent of personalized medicine, emphasizing the importance of administering different drugs to different patients. Unfortunately, today, oncologists practice the one-size-fits-all approach, or the trial-and-error strategy, prescribing the same drugs to virtually all patients with a particular type of cancer, causing more harm than good, because most of the cancer drugs have a very narrow therapeutic index, which is associated with life-threatening toxicities. Thankfully, nowadays, we have different, it starts to get induced a different approach concerning personalized medicine, which means they do some genomic testing and they have biological agents to give to these patients, which makes a tremendous progress. And as I said, I'm very glad to see that this blockbuster policy strategy starts to change towards a more personalized approach. Conventional chemotherapeutic agents such as anthracyclines may upregulate chemoresistant genes such as MDR1, which encode drug efflux protein, PGP, or cytostatic agents like vincal-alkaloids, which may activate cancer stem cells, enhancing metastatic potential. This is a very, very bad complication to cancer, because 2% of the tumor consists of cancer stem cells. These cells are quite resistant to conventional treatment. And that's why we have a very good remission in the beginning, and then there is a relapse that is very resistant to conventional treatment. This is caused by the proliferation of these cancer stem cells. Furthermore, cancer monotherapies may induce genetic and epigenetic alterations, creating chemo-radioresistance. Thus, we need personalized cancer medicine with tailored molecular targeting approaches with novel drug or gene delivery systems, which will target specifically tumor cells, circumventing biological milieu interactions. Cancer is a disease driven by progressive genetic and epigenetic aberrations, which exist as global alterations in chromatin packaging and specific promoters which influence the transcription of associated genes. Epigenetics, which is a term that is derived from the Greek epi, which means upon, and genetics, involves mitotically and meiotically heritable alterations in gene expression, which are not coded in the underlying DNA sequence. Environmental factors can influence tremendously the epigenome of each patient. Thus, in contrast to genetic alterations, the epigenetic modifications are reversible in tumor cells, favoring exploitation for drug targeting.
Excuse me, John. Did you want to share your screen? Did you want to share your screen?
Why? Can you see clearly?
No, no. I think you fell off the PowerPoint. You need to put it back on.
Okay, I'm going to do it.
Okay.
Yeah. Something is wrong here.
Okay. So you got it before. We'll get it again.
Okay, just a minute. I apologize for it.
That's okay. It's not an easy system to use. It's a little buggy.
Screen share. I go to the screen share. I press the button. Can you see it now?
Yes, we can. Yes.
Okay.
Very
I
good.
apologize.
That's no problem.
Okay, tumor-specific epigenetic mutations or epimutations at global and gene-specific level have been exhibited for all the components of the epigenetic machinery, including DNA methylation, RNA covalent histone, and non-covalent modifications, which interact with each other, cooperating with genetic mutations for establishing tumor cell-specific phenotypes, which are characterized by high tumor cell proliferation, resistant to apoptosis after anti-cancer treatment, inhibition of cell differentiation, enhanced cellular motility, and invasion causing metastasis. Another epigenetic hallmark includes non-coding RNA, such as microRNA, which may dysregulate gene activity in cancer alone, or after interacting synergistically with the rest of epigenetic components by silencing tumor suppressor genes or activating oncogenes, both of which have been identified as the drivers of carcinogenesis and tumor progression. Approximately half of the human genomic DNA is transcribed into RNA transcripts, of which 98% is non-coding RNAs, and the remaining 2% is translated into proteins. MicroRNAs are evolutionary conserved endogenous non-coding small RNAs, which act as post-transcriptional gene regulators, leading to messenger RNA degradation or repressed reproduction. Each microRNA is able to regulate several messenger RNAs by mechanisms such as incomplete base pairing and post-transcriptional gene silencing. The other post-transcriptional pathway that controls gene expression consists of the AU-rich element, the AT degradation of messenger RNA, or AMD. We must explore the interactive interplay between abnormalities of the epigenome that consist of DNA methylation and covalent non-covalent histone modifications, and the micronome, which consists of AP microRNAs, for creating new therapeutic avenues of translational medicine towards a personalized therapeutic approach mediated by tailored molecular targeting strategies against lethal malignancies. Because the epigenome differs over an individual's lifetime due to influences by environmental chemicals, drugs, diet, and aging, we need technologically advanced microarrays for identifying epigenetic changes among different individuals, tissues, developmental stages, and cancer types. This way, under a personalized cancer medicine approach, epigenetic drugs targeting histone methyltransferases, deacetylases, demethylases, sirtuins, and methylating genes, combined with microRNA mimetics or adagomers, will enable efficient, individualized cancer treatment, using molecular targeting which can act synergistically with conventional therapies or preventing resistant mechanisms for the eradication of incurable tumor cells. Emphasis must be given on the identification of biologically relevant targets of microRNAs and other epigenetic hallmarks, and how the use of microRNA mimics or adeno-microRNAs combined with inhibitors of DNMT, HDAC, histone methyltransferases, histone demethylases, and histone acetyltransferases may interfere efficiently with key molecular pathways involved in incurable solid tumors. Thus, efficient identification of the most crucial epigenetic lesions for every cancer patient might translate them into effective personalized epigenomic medicine. Another priority is to examine viral and non-viral gene delivery systems, such as nanosomes, and how we can circumvent the blood-brain barrier and all the extracellular or intracellular barriers for achieving an efficient delivery to incurable solid tumors, such as glioblastoma multiforme, of novel epigenetic drugs, including epigenetic enzyme inhibitors, microRNA mimics, and microRNA adagomers. More analytically, similarly to cancer genes that encode proteins, the regulation of microRNA encoding genes is associated with genetic or epigenetic alterations, including deletions, amplifications, point mutations, aberrant DNA methylation, histone covalent and non-covalent modifications. Recent evidence has shown that epigenetic alterations, including deregulation of microRNAs, may correlate with certain features of malignancy, such as tumorigenesis, differentiation status, and outcome of tumor patients, and indicates that microRNAs can act as oncogenes or tumor suppressors. In the future, we must we must investigate the role of epigenetic interplay on the regulation of multiple genes which are associated with diverse biological processes such as cell proliferation and development, cell cycle progression, differentiation, survival, apoptosis, invasion, metastasis, angiogenesis, epithelial to mesenchymal transition, DNA damage repair, immune escape, chemoradiotherapy resistance, stem cell behavior, and self-renewal of incurable solid tumors, characterized by poor patient median survival despite treatment with surgery, radiation, and chemotherapy, such as glioblastoma multiforme. We must identify the emerging roles of epigenetic alterations in the hallmarks of incurable solid tumors, which will lead to a clearer understanding of the mechanisms involved in the interplay between the epigenetic constituents, which will be targeted with epigenetic drugs combined with other therapies that might cause a synthetic lethality by circumvention or of oncogene dominant effects and or re-expression of apoptotic tumor suppressor genes. Therefore, the epigenetic signature of incurable solid tumors should be used for translational approaches, including development of potential epigenetic diagnostic or prognostic biomarkers, and therapeutic approaches such as inhibitors of epigenetic enzymes, microRNA mimics, or adagomers, which might be molecularly targeted with delivery systems against cancer epigenetic lesions under a personalized basis that aim to eradicate incurable solid tumors. The most crucial research question is of how epigenetic biomarkers will lead to the development of theranostics, which consist of diagnostic testing for tailoring the correct epigenetic treatment and dose on an individualized, personalized basis against incurable solid tumors. And the next lecture will be on onco-nanotheranostics, which is the basis for diagnosis, prevention, screening, and treatment and detection of tumors. Effective personalized cancer medicine requires the discovery and development with translational medicine, which means from bench to clinic, of the most effective treatments against lethal cancers that have failed all other therapeutic options. This should be novel epigenetically based therapeutic agents combined with molecular genetic approaches and standard chemotherapy for circumvention of chemoresistance by molecular targeting of signaling pathways on an individualized basis that will be facilitated by biomarker signatures derived from pharmacoepigenomics and pharmacogenomics. This is one transmission micrograph from my cancer results exhibiting phagocytosis of apoptotic bodies by adjacent tumor cells, leading to a bystander killing effect. This is the main key point here, because what we have to do is we have to induce apoptosis. We have to find dominant oncogene effect and downregulate it, okay, so we can have susceptibility to induction of apoptosis, and we go to the D2 stage of irreversible programmed cell death, as you can see here. Here you can see the nucleus. It has been destroyed because there is an enhanced electrodensity. The cytoplasm has been disintegrated. You see the organelles, they start to be disintegrated. The electrodensity of the cytoplasm has been greatly reduced. You can see here disintegration of the whole cell. the plasma membrane has disintegrated, and so you have apoptotic bodies, which, like here, which are phagocytosed, like here, for instance, by adjacent tumor cells, and that's how the bystander killing effect takes place, because these cells also will die after they phagocytose these apoptotic bodies. Okay, so this is a chain reaction effect. So here you can see entrapment of liposomes, which they are the main components of nanomedicine, by the invadopodia due to enhanced tumoral need for cholesterol molecules due to increased membrane biogenesis, because these caused by phospholipids of high TC and also cholesterol. So they are biocompatible to our body, because all our cells, the plasma membranes of our cells, are composed of the same material, from the same compounds, molecules. So that's it for today. I don't want to tire you very much like yesterday. I just went in general lines to emphasize how important epigenomic medicine is, because these alterations are reversible. And for instance, if we have a smoker who has hyperplasia and dysplasia in his lungs, and these hyperplasia and dysplasia are related to PI3K overexpression, for instance, we can reverse that. So that's one of the things that we can use to prevent cancers. And the other thing is that we can use epigenomics to alter alterations which have been caused by lifestyle conditions. And this way we can regulate our genomic pathways which are related to cancer. And this way we can reduce apoptosis. So it's extremely significant, the epigenomic medicine, because that's why I said post-genomic era, because now we can use epigenomic medicine to reverse epigenomic alterations. So, for instance, we can demethylate and we can reactivate previously silenced tumor suppressor genes up to 100. So this way we can go to apoptosis, to the programmed cell death type 1. So also histovariants, microRNAs. So as I said, one of these non-coding RNA genes can regulate up to 1,000 of the coding RNA genes of the genome. That's it.
Very good, John. Thank you very much. All three of us really don't have a heavy genomics or cancer background, so the questions we have, won't be like some of your associates. One quick question I just want to address to the students. When you say lethal cancers, you mean like lung, pancreas, ovary, brain? Could you tell us the ones you're really targeting? The worst,
yes.
the worst cancers. You mentioned glioblastoma multiforme.
Yeah, that's one of the worst.
That's the worst. And pancreatic, is that another one?
Yes, and RCC, renal cell carcinoma also, because by the time they give a symptom, they are metastatic.
Right, right.
They worked in the body for more than 5 years, so when they present a symptom, it's too late, because we have metastasis, which is a very heterogeneous tumor then, because we have 100 different tumor types in our body.
At the end, you said something very interesting. You said the treatment may reverse lifestyle changes, like smoking, I guess.
Yes.
Like smoking, someone with lung cancer, it reverses the effects, the genetic change. So, that's what smoking does. It alters the genes of the lungs. Is that what
Yes, precisely. Yes, it does.
Okay.
It does. See, to have cancer, you need more than 5 mutations. So smoking will give you 1 mutation, but you still need 4. So that's why I said if you have a very bad lifestyle, if you don't eat properly, if you eat the wrong food, okay, with pesticides, with hormones, for instance, if you don't have a good immunity system because you don't sleep enough, because you have too much stress, because your line of work is very stressful and it exhausts you. All these things, they can work against you. If you drink a lot, if you don't, as I said, if you don't sleep enough, if you don't excrete melatonin, which is a great antioxidant. So if you live a normal life, if you have, let's say, normal conditions in your lifestyle, this is going to minimize many of the epigenetic alteration risk. And also, you have many hereditary reasons that they can work synergistically if you have a bad lifestyle. So that's how you get carcinogenesis, and then you get into cancers.
Oh, okay.
By the time
Abdallah,
you,
go ahead. I'm sorry.
yeah, by the time you detect them, it's too late, because when they have when a symptom appears, this tumor is working at least 5 years in your body before it gives you a symptom. Especially tumors like pancreatic, as you said, especially like RCC, renal cell carcinoma, and also one of the worst tumors I have experienced in my cancer research experience is glioblastoma multiforme, which is called the exterminator.
Right.
It's a very resistant to conventional treatment tumor.
Right, right. We've talked about that a lot in the surgical community. Slavin or Abdallah, do you have any questions for John?
Yes, well, I happen to have a few questions if you are available, but I would first like to really comment how grateful I am to be able to see such an expert in this field to give this presentation, because you have touched many subjects that I have heard about in my medical school and also over the Hangouts, but I never got this approach that really uses them in clinical thought. And when you said that you are analyzing the genome on every patient, so this is individualized medicine, which is really, I think, the most sophisticated type of medicine. But and also I would like to comment for a second when you said that Hippocrates, that he's your greatest doctor. It is amazing because we have a statue of Hippocrates in my medical school, but he was always a doctor from some other people. But I always felt like he's also ours. But it's amazing. He was in the same country you are in now. So can you please tell us how does this feel? I mean, what is the view of Greece medicine on Hippocrates,
Okay.
and to introduce us
Okay.
in your thoughts about this?
Okay, of course. There are many other medical disciplines like Ayurveda, for instance, 7,000 years old medical discipline, Ayurvedic medicine. There is also Chinese medicine. Okay, many other disciplines. Okay, but what Hippocrates has said that for me was let's say, the most practical to our medicine today fact is that food is your medicine and medicine is your food, because food is full of phytochemicals, okay, full of nutritional substances that can manipulate our health. Okay, so without any RNA sequencing, he could predict, without any PCR, without any HPLC, without having anything of our knowledge that we have today, because we are very lucky to have all these tools now. Okay, he could predict that food can be an extremely significant factor for someone's health. And now we have nutritional medicine, we have metabolomics, now we have nutrigenomics, and now we see that the right food with antioxidants, anti-glycated agents, with anti-methylating agents, with anti-inflammatory agents, can be used as drugs nowadays. I mean, if you have the wrong diet, no matter what treatment you take, you won't be cured. Okay, you also can prevent many diseases. So that's why, for me, he was such an important doctor, because he could see that something that was not quite clear, that food is extremely important for someone's health.
Yes.
So you can use food as treatment.
So nutrition is a part of precision medicine.
Yes.
It's a significant part. Oh, okay. I didn't know
Yes,
that.
because Hippocrates as a doctor is really considered here, and I think in the whole world, as you said, one of the most important doctor of all time. So he's really a giant of medicine, and I really admire everything that I've heard and learned about Hippocrates up to this date has amazed me. So I think he's really a great role model. I would also like just one simple question, not to drag you too long, but I would just like to ask you about glioblastoma. The work that is being done at Johns Hopkins University by Dr. Quinones Hinojosa, if you are aware of this. Can you please comment about the current researches on the glioblastoma multiforme, and how do you see this area of work today?
Okay. I was very lucky to take a postgraduate degree in University of London in nanomedicine concerning liposomes, which we have used for cancer research. And as I said, liposomes are a Trojan horse. It's something that you can use to deliver your compounds, your therapeutic compounds, to your targets. Okay, because what we have here is we have a great systemic toxicity because the compounds we use for treatment, they never reach their target, and if they do, it's just a minimal quantity. So this way we have something let's say like a cell, because the phospholipids and the cholesterol molecules on their surface are quite the same like the ones we have in our plasma membrane. So this is a very biocompatible system. And this cell, you can do anything you want. You can entrap lipophilic compounds in the acyl chains of the bilayer. You can entrap polar compounds, hydrophilic compounds between the polar heads of the bilayer. You can put DNA inside for gene therapy. You can put an antibody on the surface, so you can target specific receptors in tumor cells or any other cell you might want. And you can make this carrier hydrophilic with pegylation, which means you can circumvent biological milieu interactions. This is huge. And also you can put high TC phospholipids so they won't melt in our body temperature. So you can do anything you want with this system. Okay, this is a very old system. It's not something new. Okay, but we improve it every day. We make more things to improve it.
Yeah,
Thank
I guess you've seen a lot of changes lately. I mean, a lot of changes because of the digitalization of medicine and the delineation of a genome. And you were around before the genome was broken down, right? Back in 2001, 2002. You were heavy into research then.
I have presented a long time ago one of the first, let's say, experiments of gene therapy in Brazil. It was a congress of UICC. So at that time, I have used a liposome to have gene therapy to replace a mutated p53 with wild-type p53. Mm-hmm. And that was a nanomedical gene therapy approach. Okay, since then I have improved my systems tremendously with DNA promoters. I can do genome surgery. I can intervene but it's something you gain. This knowledge is gained by time, by experience. So every time you see how ignorant you are and how many more things you don't know. And you know, I still have results from experiments before a decade that now I have the knowledge to translate the results, to interpret the results. So it's a process that you always learn. Okay, right. It's a painstaking process. I'm saying this because of the students. You learn every day, and there are so many things we still don't know. So we have to study every day to see what other colleagues have, done. And before I go to a congress to give a lecture, big congresses, they have to pass scientific committees and plagiarism software and all those things. It means I have to study all the other things that they have been done by my colleagues, see where the problem is presently, and give a solution that has not been reported before. So it's not an easy process. It means you have to study every day, okay? See where the problem is, see what your colleagues have done, your colleagues, your peers, and from there you try to find a new solution. And in all of my work, in all of my 1,000 presentations, congresses, I had this approach, this strategy. So this way I was able to contribute towards the progress of science in oncology.
Mm-hmm. Okay, I guess you've seen improvement of tools, right, to manipulate molecules.
Yes. It's something that you always, there is improvement, okay? Yeah. That's what it is. I mean, you do something, but you never reach perfection. So you always have to experiment to find a better method.
Mm-hmm. Okay, very good. Well, John, we appreciate you coming out, and we'll get better at learning this stuff. I got to hit the books and learn about a lot of these things, and I'm sure you guys. Abdul, do you have any closing remarks for John?
I just have a comment. I'm really enjoying this. It's a quite a new topic for me that I haven't tackled before, and I'm learning quite a lot actually. So yeah, I'm looking
Very
forward to the next hangout.
Yeah, we're determined to bring this to lots of people, John, and we'll help you. We'll help you as much as we can.
Really, really interesting. It's really great. Seriously, it's.
Okay,
Yeah.
very good. Thank you, John, and we'll end the broadcast and we'll chat.
Thank you so much. Thank you.
Thank you.